Hi, we are Ryan, Cherry, Monica, and Ryan from Lab Group A of Period 5, and our topic is the replication of DNA.
Cell growth and development are important in humans. In fact, around 300 million cells are dying every minute. We need cell growth and replication in order to survive. Cells are needed because they have a wide variety of usages, like growth and repair. However, before a cell is able to divide through asexual reproduction, it must first undergo DNA replication. DNA is a polymer shaped into a double helix consisting of two strands of nucleotides held together through hydrogen bonds. This is our genetic code for life, and it carries our genetic code through complementary pairing. DNA replicates through complementary pairings as they act as a parent for each new cell.
In our model, nucleotides are represented as foam, with adenine being represented by red, thymine by yellow, guanine by green, and cytosine by blue. Additional nucleotides added by polymerase are also represented by gray.
DNA replication begins with a selected area of DNA called the replication fork. This is where we will begin our replication in the model. From the replication fork, the DNA is split into two. A helicase unwinds the double helix into two parts, with one side going 5' to 3' and the other 3' to 5'. The two strands are then aided with single-stranded binding proteins that help to stabilize the strands.
The side that is 3', 5' is called the leading strand, and it starts at binding to an active site in DNA polymerase. This is where DNA starts its replication. At the active site, nucleotides complementary to the leading strand starts to bind with their complements, thus replicating the DNA. This process is continuous and goes downstream until it reaches the end of the replication fork.
On the opposite side of the leading strand is the lagging strand. The leading and lagging strand occur at the same time. However, they have a difference in that the leading strand goes from 3' to 5', whereas the lagging strand does the opposite. It goes from 5' to 3'. However, their processes are very much alike with that both require DNA polymerase. The DNA polymerase work the same in adding nucleotides to the parent strand. Because DNA polymerase only works in a 5' to 3' unit, the process is different. It needs to add additional primers in order to start. Primers are added via another DNA polymerase function as 5', so that DNA polymerase can start to add on to complementary nucleotides. Rather than continuously binding complementary nucleotides to the single-stranded DNA, the complementary nucleotides are bonded in small portions called Okazaki fragments. Thanks for watching.